with my group members, which is my name, CATAGE Ebol, Ryan, Cosmas, Robert, Anissa, Binti, Embok, and Marcia Maikel. Today we will be doing Pratcical class 1 regarding fish dissection to detect the presence of bacteria. The materials used are fish, gloves, tissue, dissecting tray, ethanol 70%, ethanol 99%, Bunsen burner, dissecting tray, water, dropper, microscope, glass slide, cover slip, media agar, and inoculation loop. Step 1. Fish dissections. Put the fish on the dissecting tray and observe the external features of the fish such as head, caudal fin, dorsal fin, pectoral fin, eye, anus, and skin. After observing all the external features of the fish are normal. Next, the fish was dissected: liver, spleen, intestine, kidney, and brain. After observing all the internal organs of the fish in normal condition. Next, take a small sample of fish tissue. Fish tissue is placed in a bottle and added with ethanol liquid. After that, the glass bottle was tightly closed. Glass bottles are labeled with 99% ethanol. The main function of the ethanol liquid is to preserve the sample so that it does not quickly deteriorate and rot. Next, we take a small sample of the intestine and add a little distilled water. The glass slide is covered using a cover slip. The intestine sample is put under a microscope to see the presence of bacteria. The microscope lens is adjusted. We use a 40x microscope lens because we can see the bacteria clearly. Step 3. Streak plate method. Media agar to be divided into 4 parts for streaking. The lobe tip will be preheated first. The tip of the lobe is used to touch the infected organ, namely the intestinal organ. The tip of the inoculating lobe was glided back and forth across the media agar surface. Touch the previous streak and repeat the streak in the remaining quarter of the plate. After all sections have been streaked, cover the plate with parafilm and place the plate upside down in the incubator for 24 hours or 72 hours.
The gram staining process involves a series of steps designed to differentiate bacteria species based on their cell wall characteristics. This differential staining technique is essential for bacteria classification and identification, which help guide diagnosis and treatment decisions. Before we begin the gram staining procedure, let's prepare all the required materials and equipment. Make sure everything is clean, ready, and organized before you begin. After 24 hours, our bacteria culture is ready for staining. Let's begin. First, label the clean glass slide with the sample ID and date. The inoculation loop should be sterilized by heating it in a flame until red hot, and allow it to cool before touching any culture. Place a drop of sterile distilled water on a clean glass slide. Spread the mixture to form a thin, even smear on the slide. Once the smear is completely air-dried, heat fix the slide by gently passing it through a flame 2 to 3 times.
Sekali ambil cair crystal.
Crystal violet was put and keep for about 30 seconds to 1 minute. The excess stain is rinsed with distilled water. The goal is to wash off the stain without losing the fixed culture.
Ada gelang iodine.
Iodine solution is used to cover the cylinder for 60 minutes. This step is known as fixing the dye. The slide is rinsed with distilled water. Excess water from the surface is taken off. Then tilt the slide into 45 degrees and add a few drops of acetone or alcohol before rinse again with tap water. After applying the decolorize for a few seconds, the slide should be immediately rinsed with distilled water about 5 seconds to stop the reaction. Add safari stain to the slide and let it sit for about 1 minute to allow proper staining of gram-negative bacteria. Gently rinse the slide with clean distilled water to remove excess safari. The slide can also be air dry after shaking off excess water, and excess water is bolted with the bibelius paper.
Oke, cecer yang atas di gelas slide ini sudah kering. Lepas itu kita letakkan di atas Microsoft untuk kita melihat bakteria yang ada.
Observe the slide under the microscope to see the stained bacteria. Gram-positive bacteria have a thick peptidoglycan wall that retains the crystal violet iodine complex, so they appear purple. Gram-positive bacteria have a thin peptidoglycan wall, lose the complex during the decolorization, and take up safranin, appearing pink.